1C, Biology and the Study of Life. Biology is often defined by its Greek word origin, bios, which means life or living, and logos, which literally means word, but has come to mean study or science of. The previous sections of this chapter discuss the term science, the logos of biology. Now the discussion turns to the other portion, or the definition of biology living. What does it mean to be alive? 1.6. The attributes of life. Rather than presenting definitions, this study will look first at a list of the attributes of life. This list does not include all the attributes, but it does present the major characteristics of living things. Since these attributes describe a very complex phenomenon, the living condition, they necessarily contain overlapping ideas. Not everything that possesses a few of these attributes is alive. Living things must have all these attributes. Non-living things, such as machines, computers, light bulbs, crystals, and chemicals, may have one or several of these attributes, but only those things that have all of them are alive. Exhibits movement. Normally, most people think of movement as motion from one place to another. Actually, moving from place to place is locomotion. Plants, most fungi, many bacteria, and even some animals cannot carry on locomotion. Nonetheless, they are alive and do exhibit movement. Movement may be internal movement as well as locomotion. Even when you are perfectly still, blood is moving through the blood vessels. From birth to death, small structures are constantly moving inside the cells of the body, even when it is at rest. Plants move water internally, dissolve minerals, and process foods. Even a stationary single-celled organism carries on internal movement, achieves growth. Though stalagmites or buildings may be said to grow, they are non-living things and grow only in the sense that more material is added to them. But to increase the size of a brain, you cannot similarly add more brain substance directly into the skull. Living things do not grow that way. Organisms grow by assimilation, lation, the assembling of the component parts that make up their living material. Growth is achieved by an organism, not done to an organism. Growth does not always affect the size of an organism. Once an organism reaches maturity, growth is often the replacement of worn-out cells. An adult, as a rule, does not continue to grow in size, but does grow new cells and parts of cells to replace older cells until he dies. Reproduces. At one time or another during its lifespan, every normal living thing is capable of reproduction. Reproduction is the making of another organism that has characteristics and limitations similar to the original. Splitting a rock in two is not reproduction, since the two halves cannot become like the original. Some small organisms do reproduce by splitting in two, but unlike the rock, each new organism has the capability to become like its parents, just as a puppy becomes like its parents. Puppies are not half dogs. They are small dogs that have inherited a dog's characteristics and limitations. Although a puppy has been nourished by its mother's body, very little of the puppy, only the original egg and sperm cells, was actually part of the mother and father. After growing, the puppy will be similar to its parents. Reproduction of living things is not making duplicates from a mold. A mother dog is not a mold for her puppies. She and the father dog have given the puppies the genetic information necessary for them to grow into dogs. Comes from similar pre-existing life. As a result of careful observations of controlled experiments and natural conditions, biogenesis is now generally accepted. The idea that life comes from similar life is important. God created humans and all of the other kinds of organisms with the ability to reproduce after their own kind, Genesis 1-12, 21-28. Therefore, humans reproduce humans, oak trees reproduce oak trees, and cats reproduce cats. The idea that all life forms descended from a common ancestor cell that originated from non-living chemicals is contrary to the teaching of Scripture and is therefore contrary to a truly Christian worldview. It is true that no kitten will be exactly like its parents. Some kittens may be slightly larger or smaller, or have different colors, depending on the genetic information given to them by their parents. These differences, some minor and unobservable and some very noticeable, are termed variations. But variations are limited. The eventual adult size of a kitten, for example, is limited to a certain range. The genetic information inherited by a kitten, along with its diet, exercise, and physical surroundings, will determine what size within the genetic range it will attain. Has similar chemical makeup. All living things are made of the same basic chemical elements in similar compounds. Since all organisms move, grow, reproduce, and come from pre-existing life, the idea that all things have a similar chemical makeup is not surprising. The substances that living things produce contain varying amounts of carbon and are called organic chemicals. Much of the composition of an organism is living material that is constantly changing. Many organisms, however, produce substances that are non-living and often remain long after the living material has deteriorated. These non-living substances are organic but are not alive. The shell of a snail, the hair of your head, and the outer bark of a tree are examples of dead organic materials. Of course, the body of an organism that has just died can also be considered dead organic material before it decomposes. Those things that are not alive and have never been alive, such as rocks, water, steel, and glass, are considered inorganic. Is composed of cells. Every living thing is made of either a single cell or many cells. Cells, discussed in chapters 3 and 4, the units of life, are limited by the membranes they manufacture. Exhibits irritability. All living things respond somehow to various forces in their environment. Irritability is the capacity of an organism to respond to stimuli. For example, human beings are highly sensitive to light and respond not only to its intensity, direction, and color, but also to its source. An earthworm, on the other hand, does not have eyes but merely light-sensitive areas. Though human beings need to respond to what they see, an earthworm merely needs to know whether it is in the light or not. In its burrow, seeing is not important. Sensing light, however, the earthworm is aware that it is not in its protective hole and can do something about it. God designed each organism to be sensitive to those conditions in the environment that affect that organism, requires energy. All living things require a constant supply of energy. Humans obtain energy from food. Plants and some other organisms obtain their energy directly from the sun and store it for future use. Food can be defined as an organic, energy-containing substance. The energy that plants store is in the form of a food. Since many organisms, like plants, do not eat food, it is not accurate to list eating or requires food as an attribute of life. Organisms require energy, maintains a high level of organization. Being alive requires that a collection of molecules be highly organized. This organized complex of molecules forms the structures of living things. Energy is required to keep these molecules in that organized condition that permits life. As soon as energy is no longer expended to maintain the organization, the molecules fall into disorder, and the organism begins to die. Think of it this way. In part, a person is a self-assembled mass of what he has eaten. He's being held together in complex order by the energy he has obtained from his food. Faces death. All things that are alive will eventually die. This fact is true for everything from petunias to people. We may be able to avoid death for several decades, but we are not able to avoid it entirely. The saddest truth we learn from biology is that nothing stays alive forever. All that lives will die. We have gone astray from our God, and He has caused our world to go astray from us. Physical life is not some special element or compound that some objects have and others do not. Life is a condition, a state in which something exists. Life can be defined as a highly organized cellular condition that is derived from pre-existing life, that requires energy to carry on processes such as growth, movement, reproduction, and responses, and that faces death. It is more important, however, to understand what life is and to recognize it than to memorize a definition. 1.7 The study of life. Studying life has always been difficult. Analyzing the chemicals that make up a living body does not reveal what life is. Diagrams of a watch can tell an engineer how the watch works, but careful descriptions of the size, shape, color, and even chemical makeup of the various structures of living things tell little about life processes. A complete set of diagrams of the human body is simply inadequate to describe how it works. The next section discusses some tools and techniques scientists use to study living things, and some of the difficulties they experience as they study. The microscope, a tool for biological study. Although a description of an organism's structures is not adequate by itself to explain life, knowledge of the structures is essential. Unassisted human vision, however, cannot see millions of microbes or the parts of cells that make up an organism. Microscopes aid in studying these tiny structures. The first microscopes were probably made in the 1st century AD by filling glass balls with water. Glass lenses that were used to magnify structures were introduced in the Middle Ages. A single lens constitutes what is called a simple microscope. Around 1590, a Dutch eyeglass maker, Zacharias Janssen, mounted 2 lenses in a set of adjustable tubes. The lens closer to the observer's eye magnified the already enlarged image of the lens closer to the object being observed. Such a microscope, consisting of 2 lenses, or 2 sets of lenses, is called a compound microscope. Because of the distance necessary between the lenses, early compound microscopes were often over 0.6 meters or 2 feet long. The best-known microscopist is probably Anton van Leeuwenhoek, a Dutch merchant who made a hobby of grinding lenses in the early 1600s. Leeuwenhoek used simple microscopes, rather than the cumbersome compound microscopes of his day. Some of Leeuwenhoek's lenses were smaller than a pinhead, and he had to push the lenses almost into his eye in order to see anything. Leeuwenhoek made over 200 different microscopes, which had various mountings to view many types of specimens. Some of his discoveries include circulation of fluids in the tiny vessels of a living fish's tail, thousands of microbes, blood cells, sperm, and even various human tissue structures. For most of his long life, Leeuwenhoek sent his observations of very many wretched little beasties, all cavorting about very nimbly, to the Royal Society of London. Leeuwenhoek, who is called the father of microscopy, never saw an object magnified more than 200 times. Today, even the microscopes commonly used in high schools can magnify 400 times. 400 times can also be written 400 times, in which the times means times larger. The compound light microscope. The compound light microscope is commonly used in classrooms and laboratories today. It consists of 1 set of lenses to magnify an object, and another set to serve as a telescope to further enlarge the image and permit the observer to look at it from a convenient distance. In a light microscope, the 2 sets of lenses are located in the objective and the ocular, AHK-U lure, or eyepiece. Many microscopes have a revolving nosepiece that contains several objectives with different magnifications. Many of the specimens that are observed using a microscope are permanently mounted on a glass slide. If a person examines the slide carefully, he may be able to tell that the specimen is colored. Special stains that react with specific chemicals in the structures of the specimen are used. Different colors react with different chemicals, so some specimens may have various colors. Thus, various structures are easily identified in a stained specimen. When a person looks through the microscope, he sees light that has passed through the specimen on the stage, through the objective lens, through the body tube, and through the eyepiece lens. Actually, he is seeing the shadow of the specimen, since the observed light has passed through the specimen, rather than being reflected from its surface. Moving the objective lens closer to or farther from the specimen brings the image into focus. Many modern microscopes have 2 eyepieces. Some of these microscopes, especially higher-powered ones, give the same image to both eyes. Others, however, are stereoscopic microscopes that provide 2 slightly different views of the same object, similar to the images you get when looking at an object with both eyes. These slightly different views result in depth perception. A number of these stereoscopic microscopes are low-powered and use light reflected from the specimen, which is placed relatively far from the objective lens. Microscopes of this nature are frequently used for dissecting small specimens. The electron microscope. One of the most useful tools for biological investigation developed in the 20th century is the electron microscope. The electron microscope replaces light rays with electrons that are exceptionally small and that travel in a straight line. Because more electrons than light waves can pass through a given area at a given time, electron microscopes can magnify many times greater than light microscopes. Since electrons are smaller than light waves, electron microscopes have a much greater capacity for resolution. There are two basic types of electron microscopes: transmission and scanning. The transmission electron microscope, TEM, produces an image similar to a light transmission microscope. Instead of light passing through the specimen, the TM uses electrons. However, electrons do not pass through substances easily. Specimens viewed on an electron microscope must be sliced extremely thin and treated with relatively harsh chemicals to allow the electrons to pass through. The specimen in an electron microscope must also be placed in a vacuum because air molecules will affect the path of the electrons. A typical TEM can clearly resolve two dots, just 0.21 micrometers apart. Electrons cannot pass through glass lenses. Therefore, electromagnets of various charges and strengths are used to pull or push on the electrons, either condensing or separating them. The electrons are focused onto photographic plates, where they are detected by electron-sensitive devices, magnified and transferred to a screen similar to a television for viewing. Electron micrographs, the pictures produced by an electron microscope, have no color but only light and dark areas, where electrons passed or did not pass through the stained specimen. Computer technicians often add color to enhance the image and make it easier to visualize the structures. The scanning electron microscope, SEM, is used to obtain surface images of structures or organisms rather than internal structures. After the specimen is coated with an electron-dense substance, the beam of electrons moves back and forth across the surface of the specimen. The coating on the specimen emits its own electrons that are picked up by a special detector. The detector transfers the energy from the electrons to a screen, where an image of the specimen's surface is displayed. The SEM can produce images up to 20,000 times. The electron microscope can magnify from 1,500 times to 250,000 times. In other words, it starts at the highest magnification possible on a light microscope and can magnify that image about 160 times. Throughout the text are micrographs produced by light microscopes, TMs, and SEMs. Principles of light magnification. Normally, the reflection of light from the surface of an object makes the object visible to the eye. If, however, a lens is placed between the object and the eye, the light waves will be bent. The wavy patterns seen through some window glass or the apparent bend in a drinking straw placed in water are the results of light waves being bent as they pass through transparent substances. This bending of light as it passes through a substance is called refraction. The refraction of light as it passes through a curved lens either enlarges or shrinks an image. There are limits to magnification. Since only a certain amount of light reflects from or passes through a specific point at one time, no image is visible if these waves are spread too far apart. The ability to distinguish fine detail is called resolution. The unaided human eye can distinguish 2 points 0.1 millimeters apart, but points closer than that appear as a single point. If 2 points 0.1 millimeters apart are magnified 100 times, however, they will appear 10 millimeters apart. When an image is magnified, the resolution increases. There is, however, a limit as to how close 2 points can be and still appear as two individual points when magnified with a light microscope. In part, the ability of a lens to clearly separate 2 points depends on the quality of the lens. Due to the size of light waves, points too close together become distorted as they are magnified. Therefore, even the highest quality lens has resolution limits. Refraction causes the pencil to appear enlarged and bent. Variables in biological studies. Because God designed life to function in a certain range of environmental conditions, it is difficult to experiment with living things. When a person wants to measure the amounts of oxygen and hydrogen needed to produce a certain amount of water, the experiment can be repeated over and over with the same results. However, when performing experiments with living organisms, Variables are harder to identify and difficult to control. Identifying biological variables. An experiment to determine how much weight a horse can pull would be affected by many variables, including the type of horse, its age, health, and size. Using the same horse for all experiments could control some of these factors, but then fatigue and boredom would have to be considered. The results of such an experiment could not be applied to all horses, only to the horse that was tested. Thus, the results would not be workable. Suppose a student conducting an experiment for a science fair project put hamsters into two groups of 5 hamsters each. His hypothesis was that hamsters on a diet of seeds and carrots would gain weight and be healthy, while hamsters being fed cake and cookies would lose weight and become sickly. After several months, all the seed and carrot hamsters remained the same weight and appeared healthy, although most of the cake and cookie hamsters also stayed the same weight and appeared healthy. 2 of them gained a considerable amount of weight. Because the science fair was close at hand, the student began to draw conclusions about cake, cookies, and hamsters. The day of the fair, though, he found 2 litters of baby hamsters in the cake and cookie group, along with 2 considerably slimmer hamsters. His conclusions had been wrong because there was a factor he had failed to take into consideration. This story is amusing. Unfortunately, eminent qualified scientists have made the same type of mistake over and over again. All the variables involved in a living condition are almost impossible to control. Therefore, reliable experimentation on living things is difficult. The validity of biological studies. From this discussion about some of the tools and techniques biologists use to study life, one thing should be evident. Studying life itself is not easy. There are far too many variables to control. Errors can be made by the biologist in his observations. Errors can result if the specimen is atypical. Errors can be made when the techniques used are crude. To overcome these problems, a scientist testing a living organism must usually repeat his work many times with a high percentage of similar results before considering a conclusion valid. The conclusion must also agree with other scientific information about the topic before it can be considered valid. A scientist performing electron microscope observations may conclude that a certain molecule is box-shaped. Another scientist performing experiments on the chemical makeup of the same molecule may conclude that it is ball-shaped. There are several possible explanations for those contrasting results. The molecule may change shape as it is prepared for electron microscope viewing, or as it is analyzed chemically. One or both scientists may be wrong in their observations or conclusions. The molecule may be both box and ball-shaped. In any case, further study is necessary. Facets of biology, biological research techniques. Today's scientists have many sophisticated tools that allow them to observe many objects that would have been impossible to see in the past. Some of the techniques described below are used in the medical profession to aid in diagnosing diseases. Others are used primarily in researching the anatomy and function of a particular organ or structure. Radiology, X-ray technology was the first development that enabled scientists and physicians to see inside the body and was first used extensively during the First World War in treating combat injuries. X-rays pass easily through some materials, but are either absorbed or reflected by denser materials. When a structure is exposed to X-rays, those rays that pass through the structure will expose photographic film to produce an image. Thus, scientists can learn about internal structures without cutting into the person or organism. Since many structures in living things are similar in density, scientists often must use substances that make the structures denser. To view the structures of the digestive system, people are often asked to drink an X-ray dense substance, a substance which blocks X-rays. The X-ray pictures then show the digestive system's structures. Computerized axial tomography scan, CT scan. Since the 1970s, CT scanning has become the most significant medical imaging technique used to screen for and diagnose diseases. In tomography, X-rays are passed through a single area from many different angles and are picked up by a detector. The information is fed into a computer, compared, and made into a picture of the anatomical structure. A CT scan can be used in many different ways to study various internal body parts. A CT scan machine is about 100 times more sensitive than a conventional X-ray machine and does not use X-ray dense material to identify structures. The information in the computer can also be used to create a three-dimensional image of the structure. Ultrasonography. Ultrasonography, URL true su NAWG ru phi, is much like the sonar used by ships. A device releases high-frequency sound waves and then records the echoes of the sound waves as they bounce off internal structures. It is pressed directly on and then moved slowly over the surface of the body, where the anatomical structures being examined are located. The resulting data is fed into a computer that produces an image based on the speed and quality of the echo. The pictures are not as accurate or precise as CT scans or X-rays, but are quite useful in observing unborn children who can be harmed by X-rays. Ultrasound pictures are also useful in evaluating the heart, which is often difficult to observe with X-rays because heart muscle is soft tissue. Magnetic resonance. Imaging, MRI. An MRI produces incredibly clear, detailed pictures of the anatomy of organisms from any plane. Rather than using X-rays or sound waves to create an image, MRIs create a magnetic field into which the patient is placed. By altering the magnetic field with a specific radio frequency, the magnetic field in the tissue is altered. The protons of the body's water molecules align with the direction of the magnetic field. The MRI machine can detect the changes in the magnetic field, and computers translate the signals into images. Patients have to be closely screened to ensure that they do not have metal objects in their body that the MRI machine might dislodge or that might distort the image. For example, patients with pacemakers or other metallic implants, such as orthopedic devices, may not be able to be scanned safely, since a patient may be harmed by the movement of the metallic devices by the powerful magnetic forces of the MRI machine. Positron emission tomography, PET. The PET scan produces three-dimensional images or pictures of physiological activities in the body that are more detailed than CTs, ultrasound, or MRIs. A radioactive substance that emits positrons, a tracer, is injected into the organism or person. As the positrons are emitted, they collide with electrons and gamma rays are formed. Special screens detect the gamma rays and computers generate an image. PET scans are used to detect the extent of cancer and heart disease and to aid in diagnosing Alzheimer's disease. Researchers are continuing to find diagnostic uses for this technology. Cellular fractionation. Cellular fractionation, fractionation, is used to remove structures from inside a cell in sufficient quantity to be analyzed chemically. After a cell has been physically broken up and placed in a solution in a tube, it can be put in a centrifuge, centrifuge. The centrifuge spins the material at extremely high speeds. This causes the pieces to settle in layers. As the denser materials settle to the bottom of the tube, the lighter materials move toward the top. Between these extremes are many layers of cellular fragments of various densities. By taking a layer of material from the centrifuge tube and repeating the process several times, an almost pure collection of structures of a certain density can be obtained and then chemically analyzed. This process permits scientists to determine what chemicals compose cellular structures. Endoscopy. Endoscopy, and DH-SCOP, is the term for a procedure that uses a hollow tube or a fiber optic cable to look inside a structure. Often the tube will have other devices in it that permit the scientist to supply light to the dark area for greater visibility, remove specimens for biopsy or analysis, help clarify diagnoses, perform surgery, or pump air into the structure so that it can be seen more clearly. Different endoscopes are designed for looking at various structures. Some endoscopes are inserted through natural body openings, such as the mouth or nose. Others enter through incisions made by a physician or scientist. Benefits of advanced research techniques. Many scientists over the last 200 years have contributed to the advancement of research techniques, equipment, and diagnostic medicine. These innovative technologies help scientists and physicians Visualize and study the anatomy and physiology of humans and other creatures, with less cutting and loss of blood and life. These men and women, using their God-given abilities, have benefited mankind by giving earlier and more accurate diagnoses of diseases, and by developing more effective and less invasive surgical procedures and treatments, increasing the rate of patient survival and quality of life.